everyone. It's cool, I've never been on this side of the podium. This is nice. So I'll be presenting some research that I was a part of this year when I interned at this biotech company called Canada. So you can see that there in the first affiliation. And so this was a preclinical drug trial that we assessed on the zebrafish model of ALS. And so before I go into the background, I've put up links to our research paper, which was published in October, and then also some of the code that I use for the downstream analysis. So for the background, canflavin A and CBD, cannabidiol, these are compounds in cannabis, and they've been independently shown to have anti-inflammatory effects. And so this company, what they're trying to do is develop a therapeutic blend of these cannabis-derived compounds for use against neuroinflammatory conditions. And they hope that by using a blend and not individual compounds, that they would be able to capture a synergistic effect that's present in the natural plant. And so I mentioned a zebrafish model of ALS, and the way that we did that model was with a neurotoxin called EMAA. And this has been shown to induce ALS-like pathology in animals, such as spinal abnormalities and motor deficits. And then the reason why we use zebrafish is because of all the disease-associated genes in humans, 84% of them have orthologs in zebrafish. And so that was important for our differential expression analysis, which I'll get into on the next slide. So this was a general overview of our methodology. So you can see there's two parts. We had a wet lab component, and the major objective there was to assess our drug phenotypically, so observing restoration of locomotion in impaired zebrafish. And then from there we sequenced RNA, and this is where I came in. So I did a downstream bioinformatics analysis, so looking at what pathways and biological processes the drug actually affects. So the first thing was a toxicity assay. It was a trial toxicity, so developmental abnormalities. And what we found is that our drug was safe below 3 micromolar, and so for all preceding treatments, we went with 2 micromolar. And that's what you're seeing here. So this is our phenotypic analysis. So I want to draw your attention to the gray bar because that's our no treatment control. And if you contrast that with the yellow bar there where we expose them to that neurotoxin, you can see there's a significant reduction in distance moved. So that confirmed the validity of our neurodegenerative model. And then on the right, those 3 bars there are different treatments. So Adarapone is a clinically approved, FDA-approved treatment currently used in practice. 402A was our code for pure canflavin A, so one of those compounds that I talked about at the beginning. And then CNR401 was our therapeutic blend. And you can see that all 3 of those restored locomotion back to that gray bar there. And so then we proceeded with a further dose response analysis. We wanted to see at what effective dose does our compound become effective. And so what you're seeing here is on the left we have CNR401, that's our drug. On our right we have canflavin A. And the main takeaway here is that CNR401 was effective at its lowest significant dose was at 0.5 micromolar. Whereas with canflavin A, it was at 1 micromolar. So we showed superior potency, hopefully capturing some of that synergy. Okay, so then like I said, we sequenced RNA, and then we moved on to the data analysis. So after normalizing data, we looked at differential expression analysis, so between 2 different treatments. Here you're looking at our pathological group versus control, and all the colored dots there are significant genes, using those thresholds there at the top. And blue is for downregulated genes, red is for upregulated genes. But I want to draw your attention to this slide because this directly compares those 3 treatments. We have CNR401 in the middle, canflavin A on the right, and Adarapone, so that positive control on the left. And just from looking at this plot, you can see that CNR401 had the broadest transcriptomic response, and this was all dosed at 2 micromolar. So you can see how that dose response assay comes into play here, because when we hold concentration constant, you can see that we have the largest effect. And that's also represented numerically in the table down below. And so you may be asking at this point, like, here are all the genes that in blue that CNR401 affects that the other 2 treatments don't. And so you may be asking, okay, we have this broad transcriptomic effect, but how do we know that it's therapeutic? And that's why I wanted to show you guys the phenotypic assay at the bottom, because we show that we can restore locomotion, and so all of these genetic changes that we see, we can assume to be beneficial and not detrimental to the organism. So the next step that I did was ortholog mapping. So this is why we use the zebrafish model. We got to see all the top genes in the zebrafish that were significantly affected and what that maps to in the human. So this is the importance of using that as a clinical, preclinical organism. But this isn't too helpful because it's genes in isolation, and so the very last step was a functional enrichment, where we extracted differentially expressed genes into functional groups, and these are biological processes that were significantly affected. And so I just grouped these into two different tiers based on what I saw. The first tier are the direct impacts. So in red we have inflammation, that confirmed our hypothesis, but then also in orange you see extracellular matrix organization. And from looking at the literature, we understand, I'm sure many of you understand too, that these have a correlation between each other, where extracellular matrix degeneration causes inflammation, and that's a repetitive cycle. And then in tier 2, this is purple and blue, you can see that there are secondary effects. So the first one, they're calcium signaling. So motor neurons are especially vulnerable to calcium overload because they don't have a great ability to buffer it. And so this would help with excitotoxicity. And then in blue, we have steroid metabolism. I know a lot of you in this room are researching estradiol and 17-beta estradiol and other hormones. And so we think that our drug is able to improve the activity of these endogenous hormones in the brain, thereby increasing the innate organism's defense system and reducing the need for exogenous supplementation. So that was my very quick, hopefully 5 minutes overview. And if you guys have any questions, I'd love to expand on any of those slides or answer any questions.